So in the first lecture of this course, we talked about amino acids and protein synthesis and the folding that they undergo. And we talked about some of the functions of some of the proteins as they relate to the cell membrane. And today we're going to talk about another class of proteins, enzymes, and we're going to be trying to discuss them within the context of botulism. So botulism poisoning is caused by the bacteria Clostridium botulinum, and they release a toxin from when there's a bacterial infection. They form these spores, and it's typically found in bacteria that's in the soil. And so, typically the notion of telling kids not to eat dirt has actual bearing. But in addition to that, botulism poisoning can emerge from also foodborne infection. It oftentimes occurs as a result of improper storage of food or improper washing of food. And in addition to that, it can occur, the infection can occur because of an infection at a wound. But there's also one more interesting foodborne infection that occurs with botulism poisoning. Can anyone think of one example of that, keeping in mind that it's a spore-forming bacteria? Yeah? Sorry? Honey, that's exactly right. That's a big reason why children typically under 1 are not allowed to eat honey because of the potential presence of botulism, particularly with locally sourced organic honey. The symptoms of botulism poisoning. The first one is flaccid paralysis. That's the inability to contract muscles. In addition to that, as a result of that, there's difficulty with swallowing or speaking. You develop facial weaknesses as depicted in the picture on the slide. They also develop drooping eyelids, and there's also dyspnea. Does anyone know what dyspnea means? Yes. Yes, trouble breathing. The particular muscle that's involved, for some of you that may have already taken physiology, is the diaphragm. The inability to maintain that negative pressure with diaphragm contraction leads to the inability to breathe and subsequent death. But prior to fatality, the symptoms that also are involved with botulism poisoning also include nausea, vomiting, and abdominal cramps. So the prevalence of botulism. Thankfully, it's fairly rare. It's something that doesn't happen too often, thankfully. And it works via the secretion of the toxin, the botulinum toxin. And that's actually an enzyme, which is a major reason why we're going to be using this to try and explore enzyme activity. And if the name botulism or botulinum sounds familiar, it's because that's also what's used in Botox. But if you think about the symptoms that we just described for botulism poisoning, that's leveraged for Botox to take away the years and wrinkles on your face. But if you also try and look up medical journals, there are also instances of botulism poisoning as a result of Botox injections. And part of the reason that happens is because this is one of the most deadly toxins. The LD50 for an average person is 100 nanograms. Does anyone know what LD50 stands for? Yes. Exactly right. Yes, that was a key part. It's the lethal dose. If you have a sample of 100 people, it is the dose at which 50% of that group will die. And so it's incredibly deadly, and it's an enzyme which we're going to be using botulism toxin as kind of a way to better understand this new class of proteins that we're going to discuss today, enzymes. So as you saw from the pre-lecture, enzyme activity has been modeled using the Michaelis-Menten kinetics model. So again, this is a model. It's meant to try and create a simplified way to understand something that's really complex. And as with any model, there are underlying assumptions that are important for its ability to represent what we're discussing. And so it's trying to depict the Michaelis-Menten model is focusing on trying to describe the how substrate concentrations reach a saturation point. And so that saturation point is the Vmax. That's the max speed at which the speed of the reaction is occurring. And that is a characteristic of the enzyme. And in addition to that, another important feature of the model is the Michaelis constant, the Km. You determine the Km by looking at the plot and trying to figure out at what substrate concentration do we get half of the Vmax? At what substrate concentration do we get half of the max speed of the reaction? And so the Vmax is a characteristic of the enzyme that will allow you to identify the Km, and the Km is identified experimentally. And so the underlying important assumptions here are that first, that the enzyme substrate complex is at steady state. What that means is that typically the substrate concentrations are going to be fairly high. And so you reach that plateau of the Vmax, the max of the speed of the reaction. And because it's plateaued there, the rate of change is 0 there. It's at a steady state and it's not changing. So if you remember from the pre-lecture, if you think about taking the derivative of the enzyme substrate complex here, that the rate of change of the enzyme substrate complex is 0. So that's the first assumption. And the second assumption is that the substrate concentration far exceeds the enzyme concentration. And so if you remember from the pre-lecture as well, that when we have the enzyme with the substrate and you get the complex, what's happening is once you get to the product, we still have the enzyme that's regenerated, and it continues to be used. However, it's the substrate that then is turned into the product. And so because of that, we need to, a critical assumption is that the substrate concentration is incredibly high. So to test your understanding of this Michaelis-Menten model, imagine that you did an enzyme rate experiment and you got the following values for the V, for the speed of the reaction at these subsequent substrate concentrations. So from this information, take a minute to try and identify what the Km, the Michaelis constant, is based on what you have given here. Does anyone already have an answer? Yeah. Yep, that's correct. So if you remember, to define the Km, you need to know what the Vmax is. So you can also appreciate it looking at it. You can also eyeball it approximately, looking at this, that as you go from 2 to 4 to 8 to 16, that it's plateauing at just over 12. And so half of that is 6. So if you try and find a value that's approximately 6, you have 5.6 there. And so that means that the substrate concentration would be just approximately above 1. You can also chart out, but this is one instance where you can eyeball it to you. Does that make sense? All right, so we have one more exercise based on the pre-lecture material that you guys went over. So looking at these two reactions that are plotted in front of you, can you venture a guess at which reaction will go faster? I'm hearing whispers of B. Would anyone like to describe, raise their hand and say why? Yes. Correct, yeah. And so if you imagine A being a reaction at which a reaction in the absence of an enzyme, B would be in the presence of an enzyme where it's lowering the activation energy and mediating the catalysis of the reaction. And hypothetically, if it was the same reaction, A would be in the absence of an enzyme, B would be in the presence of an enzyme, because it's lowering the activation energy for that reaction to occur.
So if it lowers the activation energy, is it also lowering the energy of the reactant itself?
Is it lowering? Say that question again.
Because it shows that the energy of the reactants is lower
Mm-hmm.
in the start of the reaction. Is that also what happens, or is it just?
Not necessarily. So in that example, those are just two completely different reactions in an effort to try and illustrate the what activation energy permits. And I was just saying that in an example, that's what an enzyme is able to do, but that's not speaking on the activation level of the products. Okay. So one of the important things to keep in mind is that there are also competitive inhibitors of enzymes. These competitive inhibitors, much like drugs like Cipro and anti-HIV drugs, will decrease the enzyme reaction. And we have two classes of inhibitors, competitive and non-competitive. For the competitive inhibitors, the way they act is by actually binding at the active site of the enzyme. And so typically what we have is the substrate that binds to the enzyme active site there. But with the presence of a competitive inhibitor, it'll actually compete with the substrate for that active site and block it. And so what's important to remember, again, is that Vmax is a quality of the enzyme. And so if you think about in the presence of a competitive inhibitor, again, it competes for the active site with the substrate. What will happen to the speed of the reaction if you push the substrate concentration high enough? Show of hands. What'll happen? So with raising your hand, can anyone describe to me what they think will happen if you eventually just increase the substrate concentration? Yeah. Do you think it'll differ from the reaction curve in the absence of the inhibitor? So if you think about it, what'll happen is we have a traditional reaction curve. I'm gonna pick a different color. Forgive me. That's better. So we have a Vmax here in the absence of a competitive inhibitor. And if you think about it, the competitive inhibitor is trying to compete with the substrate for the active site. Yes?
Oh, I was going to say, I think the slope will be like less steep, but
that's exactly right. You took the words out of my mouth. So remember, one of the assumptions for the Michaelis-Menten model is that the substrate concentration is incredibly high. So what will first happen is at these lower substrate concentrations, the competitive inhibitor will be blocking the ability for the reaction to occur and blocking the ability for the substrate to get access to the active site. And so what will happen is the slope before reaching Vmax, the slope will be lower. But eventually over time, because the assumption is that the substrate concentration is so high, is that it will reach Vmax. So if So if it reaches Vmax, but the slope is lower, what does this do to the Michaelis constant in the presence of the inhibitor in this reaction? Exactly right. And so you'll need more, it'll change it to become a higher concentration to reach but it'll require a higher concentration in the presence of the inhibitor. And so you can call this Km prime maybe. And so if it still reaches Vmax eventually, when you get high enough substrate concentrations, what does this tell us about the inhibitor? About its performance? Yeah. What'll happen if you think about it, as we increase the substrate concentration, what'll happen to the efficacy or the effect of the inhibitor? I heard a whisper, but I'm not sure who said it. It goes down, yeah. So for competitive inhibitors, if you increase the substrate concentration high enough, it fails the inhibition fails because it's eventually going to reach Vmax with a high enough substrate concentration. I'll stop there for a moment. Does anyone have any questions? Three hands went up. Lauren. Yeah, because if you think about it, at a lower concentration, if you imagine, depending on the dose of the inhibitor, if you imagine that there's potentially like equal numbers of competitive inhibitor molecules and substrate molecules, that'll have a specific effective inhibition. But as you increase the concentration of the substrate, all of a sudden there's more substrate being able to outcompete the inhibitor for the active site, for the reaction to occur. Does that make sense? Yeah. Do you have a question? We'll get to that in a moment. Good question. I don't want you to scoop me. Do you have a question? All right. Yes. Yeah, I should have used different colors, or I should have shaded it. So I'll do this. With no inhibitor, because it was able to reach Vmax faster in the absence of an inhibitor. But with a competitive inhibitor, it slows down, but when you get to high enough concentrations, it'll actually still reach Vmax. Yes. So the constant will increase, meaning that so Michaelis constant concentration of a substrate at which you reach half Vmax. And so if you, in the presence of a competitive inhibitor, you'll need more substrate concentration. So the Km, the constant will be higher. These are all great questions. All right. So to go to the question I got a moment ago about non-competitive inhibitors, non-competitive inhibitors will actually not bind to the active site. They'll bind somewhere else that's referred to as the allosteric site. The allosteric site, or allosteric, typically means action from a distance, or put otherwise, it's able to alter the active site from another location on the enzyme. And so in this situation, we have the substrate to the active site in the absence of a non-competitive inhibitor, and it yield a product. But when a non-competitive inhibitor gets to the allosteric site, again, acting on another part of the enzyme, it's able to have a subsequent consequence of altering the conformation of the active site. So beforehand, where we had the triangle protein being cleaved by the enzyme, now the non-competitive inhibitor binds to the allosteric site and changes the conformation of the active site to be a little more spherical. This is a really reductive way of looking at it, but does a good job, I think, of trying to communicate the point. So again, to repeat again, the Vmax is a characteristic or a quality of the enzyme. So if a non-competitive inhibitor is binding to the allosteric site and changing the conformation of the active site, what will happen to the speed of the reaction to V as we push the concentration high enough? Go ahead. That's exactly right. That was really well put. Like we've said a few times now, the Vmax is a quality or a characteristic of the enzyme. And as she put so well, we're now changing the active site of the enzyme. And as a result of that, so if the solid line is the original reaction in the absence of the non-competitive inhibitor, now what will end up happening is we have a new Vmax here. We can call this Vmax prime now, in the absence of a non-competitive inhibitor. I'll just call that NCI. It's changing the ability for the enzyme to catalyze the reaction now, because it's less efficient or less effective because of the change in the conformation of the active site. Are there any questions there? Yes.
Will it have the same slope as the one without any inhibitor?
So for my illustration, I tried to separate it just so you could see a little bit more easily. As the slide says, that a theoretically perfect non-competitive inhibitor will only change the Vmax. But again, this is only a model, and so typically you're probably going to see a change in this. You'll probably see a change in the slope in a real world experiment. I think I saw another hand up a moment ago. Yeah. It's an excellent question. Do you have any guesses as to why that might be happening?
So could like the allosteric site just be like unleverables where substrate could land, or like if it shuts off one spot?
So you're actually getting to something we're going to talk about a little bit later, but to try and steer you in the right direction, think about, so again, an enzyme is a protein, and the substrate that it's binding to is a protein as well. And so think about, or typically a protein. So think about and the allosteric site is the allosteric site of the non-competitive inhibitor where it's binding is altering the conformation of the active site. So think about, going back to the first lecture, think about what promotes changes in a protein's conformation. What are the kinds of things that alter a protein's actual like 3D physical conformation? And with that information, how do you think potentially could the allosteric site now be changing the active site? No worries. Does anyone else want to jump in? Yeah. Yeah, so you're getting to the right spot. So thinking about ionic bonding and hydrogen bonds and how now it's altering the conformation, and the conformation that's now more energetically favorable based on what's happening at the allosteric site. But to give you credit, that was a great question. Yes.
Can it be, okay, it just inhibits like for enzyme A, but can it be like activated for another?
If I understand your question, to make sure I'm understanding your question, are you asking at the allosteric site, we're talking about inhibition. Are you asking, can it enhance the enzyme's activity?
Yes, like for enzyme A, it changes, it inhibits, but for enzyme B, can it be like the reverse? It's basically,
Yeah. So we're actually going to get to that in a moment. So the allosteric site that we've been looking at, we've only been talking about inhibition. Allosteric regulation can also involve enhancement. That there could be molecules that are binding to the allosteric site of an enzyme that subsequently induce a change in the conformation of the active site, but will actually make it a more efficient enzyme at catalyzing the reaction. Is that, that's how I understood your question, is that correct? Okay, very good question, because you scooped my next slide. And so, again, so to zoom out, we talked about inhibition only. Again, to take a step back, there's allosteric regulation that can either enhance or inhibit the efficiency or the speed at which the enzyme is catalyzing the reaction. And the way it does this is that it's changing the conformation at the active site. Allosteric or allostery is activity or action from a distance. And so if you look at the graphic to the bottom right, you can appreciate that we have an enzyme that has typically enzymes will have two domains. They'll have a catalytic domain, which is where the active site is, and a regulatory domain, which is where the allosteric site is. And so the allosteric site, denoted by R, when empty, the catalytic site will bind to the substrate and you'll yield the products. In the case of allosteric inhibition, you'll have an inhibitor that binds to the allosteric site and inhibits the ability for the reaction to be catalyzed. And so when we say domains here, it's really important because it can be confusing when we're using different nomenclature when we talk about subunits versus domains. And so I want to take a quick step back to try and to help try and create clarity on what we mean on domains versus subunits. Domains are typically structurally in distinct regions of a single protein. So it's a single polypeptide chain. And so if we think back to the first lecture, thinking about structures, primary versus secondary versus tertiary versus quaternary, if we're looking at a single polypeptide chain here, what degree of structure are we looking at here? Not primary, because primary is just the listing out of the amino acid sequence. Yes. Not secondary, because again, we're looking at that would be the alpha helix and the beta sheets. Yes. Tertiary, yes, because we're looking at one single polypeptide chain here. And subunits are typically separate proteins, separate polypeptide chains that are stuck together and work together for the function of that protein. So if we are looking at a single polypeptide chain to the left, that's a tertiary structure. When it's two subunits that come together, what structure are we looking at there? Quaternary, correct. Can anyone remember an example of a protein that we discussed that has multiple subunits that come together? Hemoglobin. There you go. Well done, guys. And so turning back to the botulinum toxin, we have three domains here. We have the catalytic domain, the translocation domain, and the receptor binding domain. What's important to clarify here is that there's also labels of the light chain and the heavy chain. That's not a classification that's important for you all to remember. That sometimes, I think we talked about this in a couple of lectures now, that sometimes the way things are named or the nomenclature for different genes or proteins can be a little bit confusing. The way the explanation for light chain versus heavy chain here is because when this enzyme was first read on a Western blot, which helps you try and detect proteins and try and look at their molecular weight, what ended up happening was that there was two different chains or two different bands that popped up. And so that happens to a lot of different proteins where you'll see a light chain and heavy chain, and that's because of how it was detected and what was identified. And so for what's important for you all to remember here is that for the botulinum toxin, we have three different domains that are part of a single polypeptide chain here. So turning back to allosteric regulation, some of the different ways that happens is by the adding and removing of different groups, like phosphate groups that kinases do. Kinases will add, this is a really important enzyme to keep in mind because it's when we look at cell signaling, when we look at signal transduction and subsequent things that lead to gene transcription, kinases are what add phosphate groups from ATP, adenosine triphosphate, will lead to downstream other phosphorylation events. And another enzyme that will remove phosphate groups are phosphatases. And so kinases and phosphatases act in balance with one another, turning things, for lack of a better phrase, on and off. And if you think about it, the addition of a phosphate group will actually change the charge of whatever protein has just been phosphorylated. And as a result of that, it'll undergo a conformation that'll then permit it to now bind and interact with other proteins that would not have otherwise happened without the phosphate group, because that phosphate group is negatively charged and it's changing the conformation of that protein. And so we have kinase X that is now phosphorylating or adding a phosphate group. So it's really easy for me to forget this. Phosphorylation is just the addition of a phosphate group to a protein. And so now enzyme X has had a phosphate group added, or it's been phosphorylated. And typically, the amino acid residues that undergo phosphorylation are serine, threonine, and tyrosine, forgive me. And most often, you'll hear about serine phosphorylation, and we'll talk about that a little bit later in this lecture. Threonine and tyrosine are typically not, tyrosine phosphorylation happens as well. Threonine is less, happens to a lesser degree than the others. And there are specific kinases that only will phosphorylate a tyrosine residue. We call those tyrosine kinases. We also have allosteric regulation that will cleave bonds. And so we talked about this in the first lecture. Whenever there's the prefix of pro on a protein, it typically means the immature form or the precursor form of whatever that protein is. So a proenzyme will be converted to an enzyme. It'll be clipped and processed to remove the premature, the pro cap, for lack of a better phrase, to become an active enzyme. A really good example of this is the blood clotting cascade. That is a series of enzymes that are activating downstream enzymes that then lead to prothrombin becoming thrombin, that then cleaves fibrinogen to fibrin to form a fibrin clot during clotting. And so that's a really great example of proenzymes or zymogens becoming active enzymes. And so another example of allosteric regulation is the association and dissociation of subunits. So remember, subunits, again, are two different polypeptide chains that are coming together. And an example of this, I think I mentioned this last class, is an example of receptors binding to ligands here. So if this is a very crude, simplified cell membrane, but typically what will happen is we'll have two receptor subunits that when bound to a ligand will dimerize together. And what ends up happening is that there are tyrosine kinases here on the C terminus that when they come together, when they associate together, will activate the tyrosine kinases on the intracellular, the C terminus part of the protein, and will lead to downstream other proteins being phosphorylated. And so that's just an example of proteins when they can become associated or dissociated during allosteric regulation. Because again, it's binding at the surface here, but the kinase is down below. And cooperativity. This is something we're going to take a second to try and discuss. This actually goes back to a question someone else answered, or a question that someone else asked about how the ability of a substrate to then, when cleaved, go back and alter an enzyme's activity. In cooperativity, so what happens here is that when the catalyzed reaction occurs, in cooperativity, the product that forms can actually go back and alter and enhance the efficiency of the reaction and speed it up. And so this will not obey traditional Michaelis-Menten kinetics. And so what this leads to is a modified Michaelis-Menten equation that then includes the presence of an exponent coefficient called the Hill coefficient. And so what's important to know about the Hill coefficient is that it is meant to, it's determined experimentally. And what it does is it helps to try and include the effect of the product on the speed of the reaction. So if you look at the figure to the bottom right here, as the Hill's coefficient or n goes up, the speed of the reaction accelerates. So if you look at when it's 1 versus 2 versus 4, it's not always just a clean integer. Rarely is that the case in science. It can be a long decimated number. But as the Hill coefficient goes up, at the same substrate concentration, as the Hill coefficient goes up, the speed of the reaction is accelerated. And so this is a cooperativity where the product, in some cases, can help accelerate the speed of the reaction. An example of cooperativity is with glycogen synthase kinase 3. As I mentioned earlier, sometimes the nomenclature in science for proteins is not great. For example, we have something called MAP kinase, and there was a kinase discovered that acts on that. So we call that MAP kinase. And then we discovered a kinase that acts on that second one. So we appropriately named it MAP kinase. And so in this case, the way this works is that glycogen synthase kinase 3 will act on glycogen synthase, which then will act on glycogen synthase will then act to promote the synthesis of glycogen. And so here, what happens is there's a specific motif for glycogen synthase kinase 3, and that's usually a serine, and the X denotes any amino acid after that. So 3 any amino acids, and then a phosphorylated serine that follows that. That motif is the site at which GSK3 will act. And so in the figure, you can see that there's a kinase CK2 that acts on that first serine that's activated, and then GSK3 will then observe and see the serine XXX phosphorylated serine and mediate the reaction, and then subsequent phosphorylated serines are detected, where it continues to act and cleave glycogen synthase. That's meant to be an example of cooperativity where the product mediates the reaction here. Yes. Okay, so to take a second, so motif typically refers to an amino acid sequence that we're looking at. And so here, the motif that GSK3 will act on is serine, any 3 amino acids, and then a phosphorylated serine. And so on glycogen synthase here, for example, CK2 is a kinase that phosphorylates a serine here. Then GSK3 will detect, okay, there's a serine, any amino acid, any amino acid, any amino acid, phosphorylated serine. And then GSK3 will phosphorylate this serine. Well, then the product here is still glycogen synthase. It'll see, okay, well, we just phosphorylated this. The product is now serine XXX phosphoserine. Okay, well, now we're going to go ahead and phosphorylate this. And so it's seeing what it's done to glycogen synthase, the product, and it's able to go back and continuing to act on the product. And so this is an example of cooperativity where, again, the product will come back and be able to influence and accelerate the reaction. Are there any questions? All right, so I believe this is in the pre-lecture, to try and give you a little bit of a background for how this all applies to botulism. This is a brief video that tries to explain the mechanism of neurotransmission.
Communication between two neurons begins when an electrical impulse called an action potential travels along the axon of a presynaptic neuron toward the axon terminal. The action potential cannot cross the synaptic space. When it reaches the axon terminal, it causes membranous sacs called vesicles to move toward the membrane of the axon terminal. The membrane of the vesicle fuses with the membrane of the axon terminal, enabling the vesicle to release its contents into the synaptic space. The molecules released from the vesicles are chemicals called neurotransmitters. They drift across the synaptic space and bind to special proteins called receptors on the postsynaptic neuron. The binding of a neurotransmitter to its receptor can trigger an action potential in the postsynaptic neuron. That electrical signal then moves toward the cell body of the postsynaptic neuron. Now that the neurotransmitter has relayed its message, it releases from the receptor into the synaptic space. Some of the neurotransmitter is degraded by enzymes in the synaptic space, and some of the neurotransmitter is carried back into the presynaptic neuron through transporter proteins. The neurotransmitter that is taken back up into the presynaptic neuron is then repackaged into vesicles that can be released the next time an action potential reaches the axon terminal. The entire process repeats each time an action potential reaches the axon terminal of the presynaptic neuron.
All right, so this is going to relate back to botulism poisoning. And I want to try and take a moment to try and focus your attention towards these secretory vesicles that emerge during neurotransmission. These, as we discussed in last lecture, there are different compartments of a cell that possess a membrane, and each membrane will have specific specific molecule concentrations that are going up, or specific compartmentalization for the sake of that organelle's function. For the secretory vesicle here, its membrane will want to try and squeeze out any water molecules to try and fuse with the cell membrane. But this is a really high energy event that's facilitated by proteins that are physically grabbing onto each other from the secretory vesicle and the cell membrane, permitting the release of its contents, in this case, the neurotransmitter. And so to give you a sense of what secretory synaptic vesicles actually look like here, they are littered with tons of proteins across the surface of this vesicle. And I want to draw your attention to synaptobrevin, because that is a protein at which that the botulinum toxin targets. So zooming in a little bit on how vesicles will fuse with the cell membrane, there are snare proteins that facilitate the binding of the vesicle membrane with the cell membrane. And more specifically, there are V snares for the vesicle and T snares for the target, in this case, which is the membrane. And so each of these snare proteins, you can imagine, to try and remember two things, is that these snare proteins have specificity, that the V snares are looking for the T snares, for lack of a better phrase. These secretory vesicles, these synaptic vesicles, if you can imagine, as they are moving through, in this case, the axon region of a neuron, they're going to be bumping into other synaptic vesicles. They're going to be bumping into other organelles. And they are looking for T snare to bind with to facilitate the release of the neurotransmitter into the synapse. And the next thing to try and remember is that this is occurring, the way in which the V snare and the T snare are mediating this is that they'll actually come together. If you go from the top picture to the bottom, they'll actually, once they come in contact with one another, they'll begin to coil around each other and create these coil-coil interactions. If you can imagine having a thick strand of rope, and if you start coiling it, what will end up happening is it'll kink and it will shorten the distance all of a sudden. And that's actually what's happening between the V snare and the T snare proteins, where they're shortening the distance, squeezing out water molecules, because that's going to inhibit the fusion of these two membranes. And this coil-coil interaction is then permitting the release of the contents of the synaptic vesicle. And synaptobrevin is a V snare that's found on these vesicles. And again, as I mentioned earlier, botulinum toxin is the enzyme that acts on and cuts synaptobrevin. So the way this botulism poisoning happens is that, as we mentioned earlier, there's a bacterial infection And oftentimes, with the exception of wound-mediated infection, you know, a lot of the bacterium is consumed, and so the bacteria ends up in the intestines, and it'll actually multiply in the gut, and it'll begin to release the toxin. Now, what's really fascinating here is that the toxin, this is not easy. The toxin transports across the intestinal epithelium. Typically we have typically there are cells that will bind to their extracellular environment and bind to proteins and pull and tug. But then there's also the epithelium, which is a lining across our mucosal barrier, and they have tight cell-to-cell junctions. And so what ends up happening here, and it's I actually tried to look this up because historically, it was not clear how botulinum toxin crosses the intestinal epithelium. There's actually cells, to also try and clarify, our intestinal barrier is not just a single cell lining the entire thing, but there's an incredible diversity of cells that are present among our intestinal epithelium. And among them, I think it's called M cells. That's a site where there's a smaller amount of mucosa or of mucus creating the barrier. And it's hypothesized that it's at that cell population where the botulinum toxin can actually get through and cross the intestinal epithelium. There's really incredible experiments now where people can, in a mouse, delete specific cells and delete a specific protein in a specific cell population. And what they saw was that when they delete the specific cell, the ability of the toxin to cross the intestinal epithelium goes away. When they taught this class, again, I'm aging myself here, 13 years ago, the scientific field did not understand or did not know how botulinum toxin was getting across the intestinal epithelium. And so this is another one of those examples where cellular molecular biology is a class of half-truths, because some things that we know and understand, and some of them are still emerging. And so this is one case where it's true. And so what ends up happening is that the toxin will cross the intestinal epithelium, find its way to a motor neuron, and it's internalized by endocytosis. So in the pre-lecture for last class, I believe, the pre-lecture talked about exocytosis, how proteins are released from a cell. Endocytosis, as you can imagine, is the internalization of something. And that's not always easy to occur, or it's especially for a toxin. And so what's important to remember that we talked about earlier is that there's a receptor binding domain, a translocation domain, and a catalytic domain of the botulinum toxin. The receptor binding domain actually targets proteins that are on the surface of a motor neuron. So if you can imagine, this toxin is going around, and once it finds a motor neuron, it's actually able to bind and catch onto the motor neuron at the receptor level. And then once that happens, it's internalized. You can also think about this with the coronavirus. The spike protein is specific for every virus, is specific for usually specific cells. And with this case, the coronavirus has a spike protein that recognizes ACE2, which is the receptor on a lot of our respiratory epithelium. And so virus, in case it's toxin, have a receptor binding domain that catches and is then internalized and endocytosed. And so what's really interesting with this case with the botulinum toxin is that it's endocytosed, and typically the pH is elevated and going up. And that's a protective situation where it wants to destroy this toxin. What's really unique about this toxin is that in the translocation domain, as the pH goes up, what ends up happening, as we discussed in the first lecture, increasing pH can change the conformation of a protein. And for the translocation domain, as the pH goes up, it changes its conformation to form this almost harpoon-like structure that pierces the wall of the endosome and is then able to be free out of the endosome within the motor neuron. And then what ends up happening is the catalytic domain. I'm skipping myself here. That's the next slide. I'm just getting excited. I think this is really cool. So the toxin escapes the vesicle because of the increasing pH, changing the conformation of the translocation domain. And the light chain, or the catalytic domain, is then cleaved and is then permitted to now start targeting synaptobrevin. The way this cleavage occurs at the catalytic domain or the light chain is mediated by zinc endopeptidases. And so there's a class of enzymes called Metalloproteases where their activity is dependent on the presence of some sort of metal ion. In this case, for botulinum toxin, the cleavage of botulinum toxin for the catalytic domain is requiring the presence of zinc. And so the endo part of the endopeptidase means that it acts in the, if I go back one slide, forgive me. It acts in the middle of the protein, whereas an exopeptidase would be acting on the ends of it. Does that make sense? So a zinc endopeptidase is a protein that requires the presence of zinc to be active and to be cleaved endo in the middle of it. So now within the motor neuron, we have the catalytic domain that's present and available to find and cleave synaptobrevin. And once it cleaves synaptobrevin, vesicle fusion at the synaptic junction is no longer able to occur. It can't release neurotransmitters, and you get subsequent paralysis. So the problem with the botulinum toxin is that it keeps catalyzing the same reaction over and over and over and can survive for days. And so one of the questions that emerges as a result of that is, how much damage is it doing? And so to give you a bit of an exercise, does anyone remember what turnover rate means from your pre-lecture? Yes. So can you say that a little louder for me? Sorry, I couldn't hear you. Yes, exactly. It's at the saturation point, at the Vmax, how many reactions are occurring per unit of time for one single molecule. So I want to give you all 1 or 2 minutes to use what we have here, that if for a botulinum toxin, it has a Vmax of 14 micromolar per minute, and that's with 100 nanomolar of the enzyme. And so what is the turnover rate for botulinum toxin? You can take a minute or two to calculate that. So while you're calculating, just to remind you, this is at the saturation point, and we have the Vmax. We have the speed of the reaction at that point. It's 14 micromolar per minute. And so we have a concentration of the enzyme. So what's the speed? How many reactions are occurring per second or per unit of time within one molecule? And this is fairly similar to one of the questions or exercises in the pre-lecture as well. Does anyone have an answer? The question might sound complicated, but it's a little more, it's a simpler calculation than it might seem, if that's any help. That's exactly right. Do you want to tell us how you got that?
Basically, I just did functional analysis. So I know I want that's connected one as my results units without the so I looked at what I found. It started at 14 micromolar per minute. Because we know we have 100 nanomolar of the enzyme, and we want it for one enzyme, and then multiply by nanomolars for the product. I mean that's in terms of electrons, I converted nanomolars, and then I divide by the and then I get reactions.
Exactly right. So we have this is different from the exercise in the pre-lecture, because in the pre-lecture we had a Vmax that was in picomoles per second microgram of enzyme. But in this case, we have a certain amount of enzyme, and it's in its molar concentration, so it is already accounting for the molecular weight. And so in this case, because the amount and the Vmax are nearly at the same units, just a different scale from micromolar to nanomolar, you can just divide by the amount of enzyme that we have using its molar concentration. And typically we want the turnover rate at a per second scale. A matter of converting it from minutes to seconds and dividing by the amount of enzyme that we have, that gives us the number of reactions that happen per second for one molecule of botulinum toxin. And so to think about the effect of botulinum toxin and what it targets, what other information is needed to appreciate how damaging a single molecule of botulinum toxin is. If you do, yes. That's a good thought. That's not what I was looking for, but that's actually a great point. So remember, it targets synaptobrevin. And so it's important to think about how many synaptobrevin protein molecules are on a synaptic vesicle. So in this case, we have approximately 70 synaptobrevins per single vesicle. And within a single motor neuron, we have approximately 150 vesicles within a single motor neuron. And so if you multiply that out, knowing that we have 2.33 reactions per second with a single molecule, what ends up happening is that over the course of a few hours, one single botulinum toxin can destroy a motor neuron. And so treatment. And so there are, if you can imagine, there are antitoxins that are meant to target botulinum toxin. But as we described earlier, the toxin crosses the intestinal epithelium and finds its way to the motor neuron. And once it gets to the motor neuron, it's hard to stop. It's hard to stop that toxin molecule once it's entered the motor neuron. And so these antitoxins are meant to disable the toxins that have not yet entered the motor neuron. We also have antibiotics. As I mentioned earlier, the toxin is derived from the bacterium, and so we can use antibiotics to try and destroy the bacterial infection. But once enough motor neurons have been destroyed, and respiratory and dyspnea settles in, one of the last methods of treatment is respiratory support. Here we have a couple of professors at UVA whose research deals with bacterial toxins and membrane fusion as well. Are there any questions about any part of the lecture or what we just recently covered? Yes.
I was just wondering, is there any way to make this competitive inhibitor?
So typically, I don't know off the top of my head. I'll look into that. That's a good question. Are there any other questions? All right. That concludes the lecture. Thanks for being here, and you guys have a good day.